Hello, my name is Heather Dick, and today I'll be summarizing the molecular basis of cancer for my Biomedical Sciences 500 course. Cancer is quite a complex topic, so I'll be going over just a few of the main components and topics. While there is still quite a bit that is unknown when it comes to cancer, scientists have made great strides over the last several decades in terms of research. So first, let's start by discussing what is cancer. Cancer is, in the simplest definition, the growth of abnormal cells. These cells have the potential to proliferate rapidly and potentially spread to other parts of the body. Cancer is a disease that can manifest pretty much anywhere in the body. And in the United States alone, 1 in every 3 people will develop some form of cancer in their lifetime. Your risk of cancer increases with age, poor lifestyle, if you get a bacterial or viral infection that is linked to an increased likelihood of cancer, or if you have certain chemical exposures or radiation. And lastly, if you have a family history of certain types of cancer, it can increase the probability that you will also get cancer. So, scientists have come up with 3 models for cancer, and those are through infectious agents, environmental carcinogens, and um, epigenetics. So the first one being infectious agents, it just means the cancer is a result of cancer-causing viruses that transform normal cells into malignant tumor cells. Environmental carcinogens are just carcinogens and mutagens that cause cancer. And lastly, for this slide is epigenetics. So basically, abnormal cell differentiation, potentially in development, that leads to tumor progression. So we'll go into more detail about those 3 models in the next few slides, but first, let's discuss carcinogenesis. It is the development of cancer through a multi-step process. So first, the initiation of carcinogenesis is involves one or more changes to an important regulatory pathway, either spontaneously or from carcinogenic exposure. Second is proliferation of those transformed cells as a result of a change in one of those regulatory pathways. And third, the progression requires genomic instability in order to cause more mutations. And as more mutations occur, it increases the invasiveness of the cancer. Next, we're going to talk about infectious agents. Infectious agents are microorganisms that can enter your body in the form of bacteria, parasites, viruses, fungi, and they result in an infection that releases toxins throughout the host or body. Many infectious agents have, through research, been linked to an increased likelihood of certain cancers. Within infectious agents as a cause for cancer, there are 3 potential cancer-causing mechanisms by which this occurs. The first mechanism is the result of the body's inflammatory response to fight the infection caused by the infectious agent. So long-lasting inflammation can cause the creation of reactive oxygen species and reactive nitrogen species at wherever the infection site is. The presence of these reactive nitrogen and oxygen species can actually cause oxidative stress and begin to damage the surrounding cells and the DNA and the membrane proteins around that area. The high free radical environment that this creates increases the probability of genetic mutations and then the eventual proliferation of these mutations and eventually or potentially cancer. The second potential cancer provoking mechanism is through the inactivation of tumor suppressor genes or the activation of the oncogene pathway. These both would be the result of the incorporation of the infectious agent into the genetic information of the cell. The third potential cancer promoting mechanism has to do with the suppression of the immune system and the lack of the body to recognize that there is an invader. So the body fails to recognize the infected or transformed cells due to the influence that the infectious agent has on the host. And if the immune cells can't detect the cells, the invader cells or the infectious agent cells that have caused the mutation, they can't begin to attack those transformed cells. Next, we're going to go ahead and move on to environmental carcinogens. So we're going to go ahead and break that down. In reference to the environment, this is referring to any substance that is normally found outside of the body. A carcinogen is just any substance known to promote cancer formation. So, therefore, an environmental carcinogen is any substance normally found outside of the body that also promotes cancer formation. So, an environmental carcinogen can present itself in three different ways. It can be chemical, it can be biological, it can be physical. So first we're going to investigate the chemical carcinogen portion. That is the most prevalent of the three possibilities. And these chemical carcinogens are either hormones or fibers, which more commonly something that people can identify with is asbestos. Then we have organic compounds as well. Those are polycyclic aromatic hydrocarbons, or PAHs, which is a lot easier to say. But these are released from the burning of coal, the burning of oil, gasoline, trash, tobacco, weed. So it's a pretty common chemical carcinogen all around us. And then there's also inorganic compounds, which an example would be arsenic. So the next of those three possible environmental carcinogens are biological carcinogens. Those can be bacterial or viral in their cause. So bacterial and viral infections are considered biological carcinogens. Next, we have physical carcinogens, and this is referring to UV or ultraviolet and ionizing radiation. So that's the physical carcinogen portion of the environmental side. Next, we're going to go ahead and move on to epigenetics. So epigenetics are potentially reversible mutations, and they're typically displayed in one of two ways. So the first way is DNA methylation, which is the addition of a methyl group to DNA, which can activate or inactivate certain genes. The second of those main methods would be histone modification, and this involves the binding of epigenetic factors to the highly conserved histones, which can affect the ability of the activation of certain genes. So the disruption of epigenetic processes can lead to altered gene function and potentially malignant cellular transformation. Next, we're going to investigate the tumor microenvironment. So the ideal tumor microenvironment is well vascularized and it has a lack of tumor resistance. It has an unlimited cell proliferation potential. It's able to resist apoptosis and it's able to evade the host immune system. And these all add up and result in um, an unstable genome, which is ideal for tumor growth. There are 3 different stages to establishing a tumor microenvironment. The initiation portion is just through exposure, so whatever's going to cause that mutation in the first place. So it can be chemical, carcinogen, or infectious agents, and this will lead to reactive oxygen species, inflammation, and suppression of the immune response from the host. Next, we have promotion, and so in order to promote tumor growth, you're going to need growth factors, which are released by the tumor, and this will also stimulate angiogenesis. Next, we have metastasis, which in order for the tumor to metastasize, it needs to be able to evade the host immune system. It can't be detected while it's trying to disseminate throughout the body. So it's very important that the tumor microenvironment is one in which that is undetectable by the host immune system. Next, we're gonna investigate tumor angiogenesis, which is the the creation of new vasculature to the tumor. So, in order to stimulate angiogenesis, there's a release of VEGF, which is a growth factor that initiates this process. The increased demand by the tumor for oxygen and nutrients drives the tumor to initiate angiogenesis. So, if the tumor is unable to establish an adequate blood supply, it won't survive because it doesn't have the requirement that it needs for nutrients and oxygen. So tumor vascularization it's a lot more disorganized than it would be in the normal vascularization, and this is the result of the activation of oncogenes, the loss of tumor suppression genes, and it results in just poorly regulated angiogenic signaling within the basement membrane. So expanding on tumor invasion, so tumor invasion is the ability of the tumor cells to disrupt the basement membrane and penetrate the stroma. So it requires the loss of cell-to-cell adhesions. It needs to alterate the cell matrix adhesion and proteolysis of the extracellular matrix, and it also elicits change to the actin cytoskeleton in order to improve motility for it to disseminate within the body. The most common route for this is through the bloodstream. While there is other ways, bloodstream is the most common way, an efficient way for malignant cells to travel throughout the body. So once the tumor cell reaches the distant organ site or wherever it's planning on metastasizing, you need colonization and cell proliferation to take place in order to establish a new micrometastasis formation, which you can see in the diagram how it starts at the primary tumor and then it establishes vascularization to the tumor through angiogenesis, and then it needs to be able to survive within the bloodstream to even make it to the organ site. And then from there, it needs to again proliferate massively in order to live. Like, it needs to establish more growth. So once a new so the metastasis is the creation of a secondary or more tumor other than the primary tumor site. Next, we're going to move on to proto-oncogenes. So proto-oncogenes are genes that help the cell grow. So when there is a mutation in proto-oncogenes, which is pretty common in cancer, then they are referred to as just oncogenes, which are cancer-causing genes. Proto-oncogenes, they prevent cell differentiation, they promote cell division, and then they also inhibit cell death. Next, we're going to look more into oncogenes, which are the aforementioned cancer-causing genes. So oncogenes overexpress the proto-oncogene activity, which is exactly what they want, because oncogenes, they'll stimulate that excessive cell division, they'll inhibit cell death, prolonging the malignant cell, and then there are several different ways in which oncogenes can be activated. So oncogenes can be activated by point mutations, deletions, insertions, gene amplification, and chromosomal translocation. So for the oncogenes, how they do cause that massive cell division and proliferation, for that reason, they have become a pretty common target of many modern cancer therapies. Next, we're going to move on to tumor suppressor genes. So tumor suppressor genes inhibit abnormal cell division, they repair DNA errors, and they can initiate apoptosis. If they do find an error and are unable to fix it, they can go ahead and terminate that cell. So, tumor suppressor genes prevent the cell from dividing too rapidly. But when those tumor suppressor genes are inactivated by a mutation, then the cell division speeds up drastically, which is exactly what malignant cells want. They want that drastic proliferation. So the tumor protein 53, P53, functions as a tumor suppressor, and for that reason, it is turned off in nearly half of cancers. And it's a very essential protein for DNA repair, and it has stops in order to halt the cell cycle if it does detect damage. So for this reason, it's very beneficial for cancers to inactivate this P53 when it's trying to survive and proliferate massively. So next, we're going to talk more about the DNA repair pathways. So this includes nucleotide excision repair, mismatch repair, base excision repair. So different endogenous and exogenous agents, they induce different types of damage. So it depends on what is damaged, then we'll see, different expressions of this. So the failure of repair mechanisms results in replication errors, which in turn increases carcinogenesis, and that's why we normally see impaired DNA repair pathways in cancer. So we also see genetic instability and genome instability, which increases the risk for cancer to develop. Next, we're going to talk more about the cell cycle and the regulation it has in the normal function. So you see a loss of that cell cycle regulation in cancer. There's different DNA damage checkpoints established in order to detect cancerous cells or errors or damage in the DNA. And if it detects those, then it'll go ahead and it'll halt that cell cycle. But we often see the loss of this regulation when cancer is present. So regardless, or as we can see in the G1/S phase checkpoint, it mentions the loss of the p53 protein. So because it is no longer there because of a mutation, that checkpoint in order to see if there's DNA damage no longer exists. Next, we're going to talk about apoptosis, which is programmed cell death that can be initiated by tumor suppressor genes, as mentioned earlier. There's also cellular senescence, which is an irreversible cell cycle arrest, and it is important for preventing proliferation of cancer cells that are found to have that DNA damage. Next, we're going to talk about the role that chronic inflammation plays in cancer. So, chronic inflammation leads to the production of DNA damaging free radicals, as mentioned before, those reactive oxygen species and potentially reactive nitrogen species, as mentioned in the tumor microenvironment. The inflammation elicits a immune response similar to that you would see in an injury. So it recruits various immune cell types and stimulates angiogenesis, which is the body's attempt to fix that area of the body, treating it like an injury, but instead it is stimulating angiogenesis and providing the tumor with nutrients and oxygen to help it grow, which is why we see a link between inflammation and cancer, because it is providing tumors with their needs. So next we're going to move on and talk about biomarkers and how they play a role in your risk for cancer. So biomarkers are measurable predictors of disease incidence or outcome, and that can be measured from a process, a structure of a substance, or a substance. There are 3 different types or categories of biomarkers. The first category is biomarkers of exposure. Then we have biomarkers of susceptibility. And we also have biomarkers of effect. So, firstly, the biomarkers of exposure is the presence or detects the presence or interaction the body has with a carcinogenic substance. And these biomarkers of exposure are further sorted into if it's an internal dose or biomarker of internal dose, or if it's a biomarker of effective dose. Next, we have biomarkers of susceptibility, which indicates an individual's risk for developing cancer, but that's through variations in the process of carcinogenesis. So those variations could be a variation in excretion ability or altered metabolism that would increase an individual's risk for developing cancer. Next, we have biomarkers of effect. So they usually are early biological responses that indicate carcinogenic exposure. So next, we're just going to touch a little bit on cancer therapeutics and how the treatment of cancer is now. It varies greatly depending on which cancer is present. So chemotherapy, as you can see at the top, as it goes down the chart, you see increasing precision. Chemotherapy is more of a broad spectrum treatment when that cancer that the person's effective with doesn't have any identified specific receptors, or they've identified specific processes that can be targeted in the process of carcinogenesis. So the more that is known about the mechanism that causes that specific type of cancer, the more targeted a treatment plan can be to treat that type of cancer. So even sometimes surgical removal of an early stage cancer is the only treatment needed. So this is usually a practice seen with people who are suffering from testicular cancer, especially if it's still early on. It's stage 1, even stage 2. The survival rate for men diagnosed with testicular cancer is near 99% when it is localized to a single testicle. So typically, because the survival rate is so high, doctors want to limit the exposure of those carcinogens of chemotherapy to the patient, and will just try the simple approach of surgical removal first and see how that works out. So I have included references for each picture on the slide. You will see the reference listed at the bottom of the slide, but I've also provided a complete list of the resources that I use to complete this video. So it's feel free to pause at any time. So there's 2 pages if you would like to explore that. So thank you for watching. I have actually really enjoyed this class. 8 weeks ago, I had no idea how complex cancer was, and all of the receptors and genes and just how complex it was. And like many others, with the 1 in 3 people developing cancer in their lifetime in the US, cancer is something that has affected me personally. I've lost family and friends through the years, and so it gives me comfort in knowing that I understand the process by which cancer works. I mean, just a little bit. It gives me some comfort in understanding a little bit more. Anyways, I appreciate your time that you took to watch this video, and I would just like to leave on one of my favorite verses, which is Hebrews 11:1. It is now faith is confidence in what we hope for, and assurance about what we do not see. So, thank you so much for watching. Bye.